Hi, my name is Dr. Stacey Williams. I'm a researcher at the Institute for Socio-Ecological Research, also known as ISER Caribe. I then studied for my doctorate degree here in Puerto Rico at the Department of Marine Science, part of the University of Puerto Rico Mayagüez campus. For my doctorate degree, I researched the dispersal, the temporal and spatial dispersal of early stage larvae of Diadema, and also studied their settlement. So Diadema is a sea urchin, and if you snorkeled or dove at any reefs around the Caribbean, you might have seen these organisms. They have really long spines and they're black. So they're very important to the reef ecosystem because they are the ones that control algal abundance. And why is that important is that algae competes with a lot of benthic organisms that live on the reef. So the algae grows much quicker than, say, a coral. So if the algae are growing, they could then overgrow corals or take over space that is for possibly corals to settle, or other organisms like sponges or soft corals to settle. So they are the ones that really compete with available space on the reef. So this is kind of like a off-take of my dissertation, since I looked at the settlement of Diadema. What I'm doing now is that I'm collecting the settlers and then bringing the settlers back to lab, transporting the settlers into tanks, small tanks, and growing them out to a larger stage. And so when they get to be about 3 to 4 centimeters in width, their body size, then I will take them and transport them to the reef, where they could do their job by grazing the reef and eating algae. There's a lot of aspects of conserving and maintaining the coral reefs. So to look at herbivory, we have two main herbivores on the reefs. We have sea urchins, which the main consumer of algae is Diadema, and also parrotfish. And since Diadema died there was a mass mortality of Diadema in the early 80s all across the Caribbean. So parrotfish are the ones really controlling algal abundance on the reefs. And what the problem that we're finding now is that humans are now fishing the parrotfish and eating them. So we are really limited into herbivores on these coral reefs. So my project is to then basically build or I guess increase resilience by transplanting these Diadema on the reef and helping them reduce the algal abundance, therefore improving coral survival and improving coral settlement and other benthic organisms. One of my main goals of this project is to actually detail or to write a detailed protocol. So if somebody in the Dominican Republic or somebody on another Caribbean island wants to replicate what I'm doing here in Puerto Rico, they can. So I hope to have this protocol down set to what you should feed the urchins, how much you should feed the urchins, to what size. The problem is, the tricky part is to actually locate where these Diadema are settling. That's the tricky part. Once you locate the settlement of Diadema, then the rest of the culturing process is not so difficult. So. There have been other projects that have restored diadema to the reefs, especially in the Florida Keys. Martin Moe, he's the guru of culturing diadema. The tricky thing is that diadema have a really complex life history. So these adults spawn their eggs and sperm in the water, and then the eggs have to be fertilized in the water, and then they have a planktonic stage. And this stage could be from 30 to even 40 days in the water column. So that's really long for any type of organism in the reef. So the currents could take these plankton anywhere, basically. So they have this plankton larval stage, but then when they're in a later stage, there are certain settlement cues on the reef, and scientists don't know the exact settlement cues. Many think it could be crystallose coralline algae, others think it could be a bacterial film. It could be a mixture of things. So there has to be these settlement cues on these reefs for this plankton to then settle to the reef and then grow into an actual sea urchin. So Martin Moe, what he has been doing and in Mote Marine Lab is that he's actually spawning these urchins in lab and then rearing them through the planktonic stage. What this project is, we bypass rearing these plankton in the lab, so we don't have to worry about figuring out what the settlement cue is. We're actually collecting settlers that already have settled from the field, from the wild, and they maintain their instincts in a way that when you mess around with them and you touch them, they immediately go in a rock or in a crevice to try to hide. So they maintain their instinct to hide from predators or some external stimulation. So we're bypassing that, so we're directly collecting settlers, so we don't have to rear the plankton larvae and go through all that process. And we're also adding fish into our tanks, damselfish, so that helps these sea urchins adapt to what life is going to be like on a reef. So we're taking a couple extra steps. We're adding fish, and we're also growing these settlers to a larger stage. So when we're transplanting them to the reef, these settlers are now juveniles are going to be 3 to 4 centimeters. So they're pretty big.